So in previous lecture we have learned with the last topic centrosome and centriole. Now today we will discuss with the next topic, that is nucleus. Nucleus, and here all students don't confuse behind that word nucellus. Fine. So please you remember the difference between the meaning of these two words. Nucleus as a cell organelle. As a cell organelle described by Robert Brown. As you know that for identify mitochondria. To observe the mitochondria, we can stain with the help of the genus green. Fine. Mitochondria is stained by genus green. So to observe the mitochondria, we can stain it by the genus green. Same way, to observe nucleus, stained by acetocarmine. We can stain the nucleus with the help of the acetocarmine. Fine. In the 12th standard, we can perform the one experiment. We can take the very thin layer which is there in the onion. You know that actually onion is a tunicated bulb, and in onion the food is stored in the leaf base. So in between the leaf base, very thin layer there. So we can take the small section of this thin leaf, and then we can stain it by the acetocarmine. It is reddish in color, and you see that dark red color of observation, and we see that under the microscope. We can see the nucleus under the microscope at low power. Fine. In the 10x we can see it easily. The name of nucleus Alexander Fleming actually. And as you know that Alexander Fleming discovered the first antibiotic drug, that is penicillin, which is obtained from the Penicillium notatum fungi. It is controller or director of the cell. It is also known as controller or director of the cell. Nucleus is important for controlling the heredity, growth and metabolism. Nucleus is important for controlling, or it is important for the inheritance, growth as well as metabolism of the body, and all the activities are controlled by the nucleus. That type of the function can be was discovered by Hammerling and scientists. Hammerling scientists worked on the Acetabularia like algae, and it is the single cell largest algae. Fine, and you know that nucleus is absent in mammalian RBC. The most important line. nucleus is generally absent in the mammalian RBC and in the sieve cells or sieve tube. Actually, it is the part of the phloem, but with the exception, the nucleus is present in the RBC of such mammals like, but here exception there, camel and llama. Yes, you search the name ILAMA. Actually, I is silent there. Llama and camel both are belonging to the one family, Camelidae. So this Camelidae family, in that two animals, camel and llama, both consist the nucleus present in the RBC. Paramecium is unicellular but dikaryotic, two nucleus there. Paramecium is unicellular and dikaryotic. Unicellular means single cell and dikaryotic means two nucleuses are there. Here, the nucleus consists nuclear membrane. Nuclear membrane also known as nucleotheca. Nuclear membrane is also known as karyotheca, whereas nucleoplasm, also known as karyolymph or karyoplasm. Portion it is known as nucleus matrix or nucleoplasm. It is also known as karyolymph or karyoplasm. Whereas nucleolus it is also known as little nucleus, also known as ribosome factory. Perinucleus space there. So nuclear membrane is double layer and both the layers are parallel to each other. So in between the two layers the space is present. It is known as perinuclear space. So perinuclear space consists 10 to 50 nanometer area. Now here the nucleus consists chromatin neck. Or we say that chromatin fibers. Okay, nucleus. Actually, dear all students, 12th standard, the packaging of the DNA helix there. Actually, in the structure of the nucleus, chromosome is there. Yes, we say that. Actually, nucleus is made up of the chromosome, and this chromosome consists chromatin fibers, and this chromatin fiber, it is arranged in highly network-like structure in the form of net. But we say that chromatin fiber or chromatin material, it is made up of DNA, RNA, histone proteins. Okay, histone proteins and non-histone proteins. That's why in the nucleus, the chromosome is actually nucleoprotein. Chromosome is actually consists of nucleoprotein, and nucleoprotein in that majority nucleic acid, that is DNA as well as RNA, and along with that histone as well as non-histone protein. So here total 9 non-histone proteins are there. Sorry, histone proteins. Total 9. Okay, H1, H2A, 2 molecules, H2B, 2 molecules, H3, 2 molecules, and H4, 2 molecules. So except the only H1, H2A 2 molecules, H2B 2 molecules, H3 2 molecules, and H4 2 molecules forms histone octamer. Okay, octa means 8 molecules are there, and this histone octamer is surrounded by the 200 base pair consisting DNA. Okay, histone octamer wrapped by 200 base pair containing DNA. Okay, so one histone octamer. One histone octamer is surrounded by 200 base pair containing DNA. This structure is known as nucleosome. Known as nucleosome. And in histone protein as well as non-histone protein molecule there. So here two amino acids are present in the most abundant amount, arginine and histidine. Okay, so in the histone and non-histone protein, in both this arginine and histidine are present in the most abundant amount. Sorry, arginine and not a histidine, lysine. So, histone proteins form the histone octamer. Now you are thinking about that what is the function of the H1? H1 connects the two successive nucleosomes. H1 acts as a connection or connecting protein between two successive nucleosomes. So actually nucleosomes consist histone octamer and 200 base pair. So many nucleosomes are united together and interconnected by the H1 histone protein. Okay, in this way, long thread-like structure it is formed that it is known as chromatin. Fine. And then so many chromatin fibers are united together from the chromatin net, and then very complex chromosome is formed there. Okay. Now, so these are the total 9 functions of the total 9 histone proteins. Now you are thinking about that what is the function of the non-histone protein? DNA is connected to RNA, is connected to histone as well as other proteins also, other molecules also. So 3-dimensional structural stability that is controlled by the non-histone protein. So non-histone proteins are of two types, euchromatin and heterochromatin. Non-histone proteins euchromatin and heterochromatin. Euchromatin is able to transcript not transcripted. You know that transcription it means DNA is converted into mRNA. So euchromatin is metabolically active. Euchromatin is metabolically active and it is able to transcript, means DNA is converted into RNA, but heterochromatin is metabolically less active, which is not transcripted at all. Now, euchromatin is metabolically active, and it is able to transcript, means able to form the converted into mRNA, and then mRNA is converted into protein there, and then later on that protein forms the particular function. So now here heterochromatins heterochromatin is of two types, constitutive and facultative. Constitutive means present in all the stages of cell. Okay? Constitutive heterochromatin which is present in all the stages of the cell. And now understand this one, it is very important. Facultative, present in certain stage of cell only. Here we say that one example, bar body. Now I want to discuss with this bar body topic in detail. Bar body is one type of the extra chromosome. One type of the extra staining or extra chromosome. Okay? It is discovered by Bertram scientist. But normally I can give you the meaning. Bar body means total number of X chromosome minus 1. This is known as bar body. Bar body is very useful for human being. So bar body means total number of X chromosomes minus 1. Okay, you can easily understand this. Normal female consists one bar body. Yes, now you know that normal female consists total 2 X chromosomes. So 2 X chromosomes minus 1. So one bar body is present in normal female, in human being. Fine. And normal male consists no bar body. Why? Because normal male consists only one X type of the chromosome. Now, very interesting topic. Actually, surrounding to the embryo, amniotic fluid is present there. from this amniotic fluid, the embryo can get the frictionless movement. Along with that, amniotic fluid acts as a shock absorber. It acts as a lubricating fluid as well as shock absorber. So, at the time of the 3 to 4 months of the pregnancy, more than 3 months, okay, at the time of 3 to 4 months pregnancy or more than that, if we obtain the little bit amount of the amniotic fluid from the pregnant lady, if we obtain the little bit specimen of the amniotic fluid, then from this amniotic fluid we identify the two things. Number one, any genetic disorder, any type of the genetic disorder in the developing embryo. So if any type of the genetic disorder there, we can able to cure it, or otherwise we can abort the embryo also if incurable genetic disorder is there. Then another thing, we can identify the sex of child. How we identify the sex of child? By obtaining the number of the barcode. So if in the amniotic fluid one body present, this indicates that female embryo is present, definitely. And if no body present, so we say that male baby or male embryo is developed there. So this is about the bar body. It is the facultative heterochromatin. So I hope you understand this many topics. So here I can stop now. Fine. Remaining thing we will discuss in the next lecture.